I welcome you all in the Swayam course entitled Genetic Engineering and its Recent Advancement. Till now in this course we have studied about genome structure, we have studied about DNA, cloning, the vector that are used in cloning, transfection, and I think a little bit about gene silencing in the previous lecture. So in this lecture I will be talking about RNA induced gene silencing, and in this we will be talking about siRNA and miRNA. So before starting with the topic, let's just brush upon the main concept of the central dogma of molecular genetics. So it consists of three things: DNA replication, transcription and translation. So basically central dogma is the transfer of information from the genetic material, that is DNA, for the synthesis of mRNA and finally protein. So talking about RNA interference or RNA induced gene silencing, before starting to that, let's go back to the history, why people started studying about these kind of technologies. So everything going back to the history of Mendel or any history of genetics, these kind of studies was done to study the function of gene. Alright, so from the past there are basically two methods for the study of gene function. One is the forward genetics, another one is the reverse genetics. In forward genetics, it involves the identification of the genetic basis of the phenotype that you have. That means in forward genetics you have the phenotype, you want to study what is the gene that is responsible for that particular phenotype. Alright, you have the phenotype in reverse genetics. On the other hand, so in forward genetics, it involves identification of genetic basis of the phenotype. What you have is the phenotype, how something looks, may it be the hair color, may it be the eye color, may it be the pod shape. You want to know what is the gene that is responsible for that particular phenotype. Whereas in reverse genetics, it involves genetic manipulation or mutation of the gene so that we can study what is its effect in the resulting phenotype. Alright, in reverse genetics, you have the gene. Now you want to understand what is the function of that gene. For that, and the gene silencing that we are going to study today comes under reverse genetics. So basically, starting with RNA interference. RNA interference is basically the process in which the gene expression is inhibited by the double-stranded RNA molecules. How? By neutralizing the targeted mRNA molecule. These double-stranded RNA molecules hybridize with the mRNA and suppress gene expression. So the RNA interference is basically an evolutionary conserved mechanism which is triggered by the double-stranded RNA that uses gene's own DNA sequence to turn it off. This process is known as gene silencing. It is a basic regulatory mechanism that limits the level of transcript or the gene expression in two ways. First way, by suppressing the transcription. It is known as transcriptional gene silencing. The second mechanism is by degrading the mRNA produced. It is known as post-transcriptional gene silencing. In post-transcriptional gene silencing, either the mRNA is degraded or the translation is inhibited. That means the protein will not be synthesized. That means the gene will not be expressed. Talking about the history of gene silencing or RNA interference, to be very specific, this process was discovered by two American scientists, namely Andrew Fire and Craig G. Mello. And they discovered RNA interference in the cells of C. elegans. What they did was they introduced short segment of double-stranded RNA into the cells of C. elegans and to see how they are inhibiting or how they are affecting the expression of certain genes. For the discovery of RNA interference, these two American scientists were awarded the 2006 Nobel Prize in Physiology and Medicine. Now, understanding what exactly was the experiment which led to the discovery of RNA interference. So basically, they studied the phenotypic effect of the injection of single-stranded and double-stranded RNA molecule and specific RNA molecule which are targeting the UNC22 gene. Alright, the double-stranded and the single-stranded RNA that they took, they were targeting the specific gene UNC22. That means they were targeting the specific mRNA for UNC22. The UNC22 gene is encoding the myofilament protein. the myofilament protein in C. elegans, if it is absent, that means if the gene identifying the myofilament protein is silenced, it is not expressed, it will result in the twitching of C. elegans. So coming to the experiment, what they did was, they took three segments. First, they take the sense RNA, single-stranded sense RNA, they took the single-stranded antisense RNA, and they took double-stranded small segment of RNA. They injected these three RNA, which was specifically targeted for UNC22 mRNA. They injected these three types of RNA into the gonads of C. elegans to see the progenies. So what they observed was, when the single-stranded sense RNA was injected into the gonads of C. elegans, the progeny was absolutely normal like a wild type. Similar observation was observed for the single-stranded antisense RNA targeting the UNC22 gene. So what they did was, they did three experiments. First, they took the single-stranded sense RNA targeting the UNC22 gene. In another, they took the antisense RNA single-stranded targeting the UNC22 gene. And in the third experiment, they took double-stranded RNA small segment targeting the UNC22 gene. When I am saying targeting the UNC22 gene, that means they are targeting the mRNA basically. So in the first experiment, that is the sense RNA single-stranded, the progenies were absolutely normal like a wild type, no twitching. Similar observations were observed in case of antisense RNA single-stranded. There were no twitching in the progeny of C. elegans, that means it was similar to the wild type. On the other hand, in case of double-stranded RNA, the twitching was observed. That means the gene silencing of UNC22 gene was observed in the C. elegans gonads when double-stranded RNA was used. This observation and this experiment led to the discovery of RNA interference. Before moving to the genetic basis and the molecular mechanism of this RNA interference, there are few terminologies that we need to understand: the sense strand and the antisense strand. Okay, let's consider a double-stranded DNA. One strand of the double-stranded DNA is the sense strand. The another strand which is complementary to the sense strand is known as antisense DNA strand, which acts as a template for transcription. So when the transcription happens, the mRNA which is synthesized using the antisense DNA strand as a template is known as the sense RNA strand. After the sense RNA strand, or let's call it mRNA strand, is synthesized, the double-stranded DNA detaches itself and the mRNA is free. Now, antisense RNA strand means a strand which is complementary to the sense mRNA strand. Okay, so we need to remember these terminologies of sense and anti-strand to understand the basic mechanism and concept behind RNA interference. Before going to the RNA interference mechanism, let's talk a little bit about what exactly are these molecules. These small RNA molecules are said to be in control of gene expression, or they can control the gene expression. These small double-stranded RNA molecules, how strong? These small double-stranded RNA molecules are around 20 to 25 nucleotide long and are known to regulate the gene expression in the cytoplasm of higher plants, lower plants, animals and fungi by repressing the translation and triggering the degradation of mRNA. This form of sequence-specific post-transcriptional regulation is known as RNA interference. There is one mechanism. Another mechanism, also short RNAs act in the nucleus to alter the chromatin structure and bring about the repression of transcription, because if the chromatin structure is altered, the DNA that is there will not be able to do transcription. So mRNA will not be produced. So together, these two mechanisms, where there is post-transcriptional gene silencing and transcriptional silencing, together these two phenomena is known as RNA-induced gene silencing. So the main two types of RNA molecules that take part in RNA interference are microRNA known as miRNA and small interfering RNA or siRNA. These two mRNA molecules which take part in RNA interference are double-stranded, 20 to 25 nucleotide long. The difference in the structure, the miRNA or the microRNA has the loop, is double-stranded, it has a loop-like structure. However, small interfering RNA or siRNA is again double-stranded, but it has no loop. Talking about the basic concept, how RNA interference work. As in the experiment, as I told you, the double-stranded small fragment of RNA was working for gene silencing. So, double-stranded RNA has two strands. One is the anti-sense strand, one is the sense strand. Now, talking about the central dogma. You have a genetic material, right? You have a gene which has a genetic code for a particular mRNA and a particular protein. Via DNA, through transcription, mRNA is synthesized. Now, in this RNA interference, this antisense RNA which is targeting this particular mRNA is complementary to each other. So, this antisense RNA strand hybridizes to this synthesized mRNA and blocks the protein synthesis. Alright, either blocks the protein synthesis, or what happens after the hybridization of mRNA with the antisense strand of siRNA, this mRNA is degraded by nuclease activity. So, this is the basic concept how RNA interference works. So, the two major types of short RNA molecules, as I told in the previous slide, are siRNA, small interfering RNAs, and the microRNAs or miRNA. Both the similarities, both are short, double-stranded molecule between 20 to 25 nucleotide long. Now, we will deal with individually siRNA and miRNA. Before going to the molecular mechanism, we will talk about how this siRNA and microRNA come to the cell, how they are, वो आते कैसे हैं cells में, हमको ये समझना है. So, the siRNA or short interfering RNA are derived from a longer RNA molecule that are linear, double-stranded and located in the cytoplasm. How this long, linear, double-stranded RNA comes to the cytoplasm? It comes from the viral infection. Because of its mechanism or life cycle, it produces or synthesizes this linear double-stranded RNA molecule. In nature, these siRNA precursors, these long, linear, double-stranded RNA molecule in the cytoplasm synthesized by virus are known as siRNA precursor. They arise within the cells as a result of virus infection, as I told you. RNA interference may be also a method which cells recognize these double-stranded RNA from a foreign particle and inactivate them to protect the organism from external or internal stimuli or infection. Now, these double-stranded RNA molecules can also be introduced into the cell for research and therapeutic purposes, which are recognized by an enzyme complex known as dicer in the cytoplasm and can be cleaved into siRNA. So, basically what happens, the single-stranded structure of siRNA precursors are recognized by the dicer enzyme complex. They are cleaved into siRNA, and this is how siRNA comes to the cytoplasm. Moving ahead to microRNA. These are derived from the single-stranded RNA that are transcribed within the nucleus from the cell's own genome and that contain a double-stranded stem loop structure. What happens in the nucleus when the mRNA is synthesized via transcription? They are single-stranded. What happens sometimes? They make a loop-like structure. Within themselves, these are single-stranded RNA. Within themselves, they make a loop-like structure like this one, complementary to each other, and they have a loop-like structure. The nuclease enzyme present within the nucleus recognizes the stem loop structure of the mRNA and cleave them from the longer single-stranded RNA. These stem loop RNA fragments are then exported from the nucleus into the cytoplasm where they are miRNA precursors. Further, they are processed by the dicer complex into short, linear, double-stranded miRNA. And this is how miRNA comes to the cytoplasm. Talking about the mechanism. See how in the cytoplasm, in the nucleus, these precursors of siRNA and miRNA comes into existence, we have already talked about. The dicer molecule, the dicer enzyme complex, recognize this miRNA precursor and siRNA precursor and cleave it to make a small 20 to 25 long nucleotide long siRNA and miRNA double stranded RNA. Now they have two fate. They can either complex with RISC, RNA induced silencing complex, or they can complex with RITS, RNA induced transcription silencing complex. First fate, if the siRNA or the mRNA molecule associate themselves with the enzyme complex called as RNA induced silencing complex or RISC, what happens when they complex with RISC? They are double stranded. When they complex and they enter the RISC complex, one strand, the sense strand of the double stranded siRNA or miRNA is degraded. Now what they have is now single stranded antisense RNA RISC complex, and they are functional highly specific agent of RNA interference technology, and they seek out specific mRNA molecule which are complementary to the antisense RNA contained in the RISC. At this point, RNA interference can take one of the two pathways. If the antisense RNA in the RISC complex is perfectly complementary to the mRNA, this antisense RNA of RISC will complementally properly bind with the mRNA. But what happens in this case? The nuclease enzyme will cleave the mRNA and degrade it. If the antisense RNA within the RISC is not exactly complementary to the targeted mRNA, then it somehow binds to the mRNA because they are somehow complementary. But what will happen? The ribonuclease will not degrade that mRNA antisense complex, but this mRNA now cannot take part in the translation. So in both the case, protein will not be synthesized. This is one mechanism. Now what happens if the siRNA and the miRNA binds to the transcription silencing complex RITS, RNA induced transcription silencing complex? If they associate with RITS, again as happened in RISC, the sense strand of the double stranded miRNA or siRNA is degraded inside the complex. Now what they have is the antisense RNA along with the RITS complex. Now this complex is active and they target to the specific gene promoter or larger regions of chromatin. They will not target the mRNA. They will target a specific gene promoter or larger regions of the chromatin. After combining or maybe attaching themselves to that promoter region or the larger region of chromatin, they recruit chromatin modifying enzyme in these regions where they are attached. Now these enzymes methylate the histone proteins of the DNA and result in the heterochromatin formation. Now because of this modification by RITS complex, this results in the silencing of transcription because now the modified DNA cannot take part in the transcription. So in this case, in this mechanism, the mRNA will not at all be formed. The transcription is silenced. So in both the cases of RNA interference, either the mRNA is degraded, if that is the case, the protein synthesis will be blocked. If it complexes with the RITS, they recruit enzyme which will modify the chromatin, they modify the DNA such that this result in the silencing of the transcription. That means the DNA will not be able to participate in the transcription mechanism. So why are we studying this technology? Why this is very important? Because of the varied application it holds. Starting with the gene function study, one of the very important application of RNA interference is a very powerful tool to study the gene function. By silencing the specific gene in a controlled manner, we can identify the function of the targeted gene. That is one very important application. Next is the drug target validation. Identifying and validating potential drug targets is a very crucial step in drug discovery. This RNA interference technology can be used selectively to silence candidate gene to assess their impact on disease progression, helping the people who are working on drug discovery to prioritize target for drug development. Again, a very important application for drug discovery. Next is the functional genomics. RNA interference enables large-scale systematic analysis of gene function across entire genome. This has been instrumental in understanding the role of gene in various organisms, including humans. Next is the therapeutic application. First and foremost and very important application of RNA interference is cancer therapy. This RNA interference can be used to target and silence specific genes which are involved in cancer growth and progression, potentially leading to the development of novel cancer therapies. Similarly, for virus infection, these RNA interference can be employed to inhibit the replication of viruses by targeting their essential gene or viral DNA or RNA, let's say for example. This approach holds promise for antiviral therapy. Next, very important, neurodegenerative diseases like Parkinson's and Alzheimer's. We all know Parkinson's and Alzheimer's are very potent diseases because of which we don't have any treatments. We don't have any potential treatments. So RNA interference can be a very, very good tool to silence specific genes which are associated with Parkinson's and Alzheimer's disease. Next, similarly, the genetic disorders, RNA interference can be used to modulate the expression of genes responsible for the genetic disorders, offering potential avenue for treatment like sickle cell anemia, thalassemia, and you name it. Furthermore, in agriculture sector, for the development of functional crop improvement, RNA interference technology can be applied to enhance crop traits such as resistance to pest, diseases and environmental stresses, as well as to improve nutritional content. Similarly, to control the pest, developing RNA interference based pesticides can target specific genes in pest, reducing the need for chemical pesticides and minimizing the environment impact. Functional proteomics, RNA interference can be used to study the function of a specific protein by silencing the gene that code for them, allowing researchers to investigate protein-protein interaction and cellular pathways. The next application is stem cell research. So in stem cell research, RNA interference can be used to regulate gene expression in stem cell, allowing researchers to control the differentiation into various cell types for regenerative medicine. This regenerative medicine is a very, very important field where different types of stem cell can be used, which are modified by this technique for the generation of cell lines, which are very important for therapeutic uses. Next is the personalized medicine. This RNA interference can potentially be used for personalized therapies by targeting specific genes associated with the individual disease or genetic makeup. Personalized medicine, that means a therapy for a particular disease which is there for an individual's disease or maybe for a group of people or a group of a genetic pool. This can be used by targeting a specific gene which is responsible for the diseases which are there in available or which are there which is very much present in the group of people. And finally, vaccine development. RNA interference can be employed to enhance the effectiveness of vaccine by modulating immune responses or by targeting specific antigens. We all know, we have all been through the very tough time of COVID pandemic. Did any one of us realize or understand how the vaccine of such complicated disease which was caused by virus, how the vaccine was discovered so fast? Thanks to these kind of technologies, bioinformatics, so the vaccine can be discovered at a faster rate. So RNA interference is one such technique which can be used to, to develop vaccine for such diseases. Next application, a very important application, is functional analysis in model organisms. So RNA interference technology is widely used in model organisms like C. elegans, Drosophila, zebrafish to investigate the gene function and disease mechanism. So at laboratory level, functional analysis of the function of the gene can be analyzed in model organism. See, RNA interference is a very specific technique where you can target a very specific gene by targeting its mRNA. By silencing the gene, you can understand the function of each gene. Using this technique, a library can be made, like a library of C. elegans has already been made. We have for Drosophila, we have for zebrafish, and maybe some more lower and higher organisms. So why this technique is important for such library synthesis or say library development? Because this RNA interference technique is very, very specific to a particular gene. Next is biotechnology and bioprocess application. This RNA interference can be applied in biotechnology to improve the production of biopharmaceuticals, optimize the metabolic pathways in microorganisms for biofuel production, and to enhance protein expression in cell culture systems. Again, finally coming to the industrial sector. So this RNA interference is a very, very potent technology which can be applied to a varied group of industry which are related to biotechnology and bioprocess. We can optimize the metabolic pathways. We can tweak the metabolic pathways by silencing the genes or the pathway which are not very important, or maybe which are interfering with the yield of the required product. For example, biofuel production, the microorganisms that are used. We can tweak the pathway of the microorganism because they are lower organism. Tweaking the metabolic pathway or silencing a gene in a microorganism is very easy, and they are very well used for the biofuel production. Similarly, we can enhance the protein expression in a cell culture system. How can we enhance the protein expression of a particular protein? By the protein or the gene which is affecting the synthesis of the required protein can be silenced, which can enhance the protein expression of the required protein. So these are the major applications of the RNA interference, and this is an ever-changing field. There are so many things that are already happening in RNA interference. People are experimenting. There are so many things which are yet to be discovered. The horizon is very wide. So just revising this topic, what have we studied today? So we started with the central dogma. I made you understand what the central dogma is, and RNA interference, why I told you about central dogma, to make you understand RNA interference, where exactly it works, how it's silencing the genes. Alright, then we talked about what is RNA interference. Alright, then talking about the history. Who were the scientists that discovered the RNA interference? They got the Nobel Prize in Physiology and Medicine in 2006. We talked about that. Then we talked a very important thing, that was the experiment that was conducted which led to the discovery of RNA interference in C. elegans. Alright, then we quickly moved to the basic concept. We talked about sense RNA, antisense RNA, how this antisense RNA strand bind to the targeted mRNA and blocks the translation, or maybe degrade the mRNA. Moving on to the molecular mechanism, we talked about there are two types of basically molecules of RNA which are used in RNA interference, siRNA and miRNA, small interfering RNAs and microRNAs. We talked how they reach to the cytoplasm, how they are synthesized in the cells, how the dicer enzyme complex recognize these precursor molecules, and they led to the development of siRNA and miRNA. Then the two mechanisms where they bind to the RISC complex and led to the inhibition of translation and degradation of mRNA. That was one mechanism. Another mechanism was transcription silencing, where the siRNA molecule and the miRNA molecule were binding to the RITS complex, RNA induced transcription silencing complex, where they were binding to the chromatin, to the DNA molecule directly, or the chromatin molecule, where they were methylating the histones, and they were affecting the DNA molecule so that they are damaged, they are manipulated, and so that they cannot take part in transcription. So these were the two mechanisms that we studied of RNA interference. These mechanisms are very important to understand, because if you don't understand the mechanism, you cannot apply in the applications I told about. So we talked about a lot of applications ranging from therapeutics to plants to bioprocess industries, personalized medicine. So this was about RNA interference. So talking about how siRNA and miRNA reach to the cytoplasm, we talked about it a little bit in the previous slide. So in the cytoplasm, due to the virus infection, the double-stranded linear RNA molecule reach to the cytoplasm, and these virus molecules synthesize this double-stranded RNA because it's their genetic material as a normal cell cycle mechanism. So when they reach the cytoplasm, the double-stranded RNA molecule is recognized by the dicer enzyme complex. So this dicer enzyme complex recognized this siRNA precursor, which are the long stretch of double-stranded RNA molecule. This dicer molecule then cleave it into small 20 to 25 nucleotide long small interfering RNA molecule. This is how siRNA come to the cytoplasm. Now coming back to the miRNA or microRNA. So this microRNA is synthesized by the normal mRNA that is synthesized in the nucleotide. So when the mRNA is synthesized in the nucleus via transcription, many a times it happens this single stranded mRNA, the linear mRNA, make a loop like structure. They get complemented to each other, long strand come near to each other and they make a stem loop like structure. Now this stem loop like structure will not be able to take part in translation. So what happens, this nuclease enzyme in the nucleus recognizes this miRNA precursors or this loop like stem structures and they cleave it. After cleaning, this small miRNA precursor, which are like stem loop structure, they reach to the cytoplasm, they are transported to the cytoplasm. When they reach the cytoplasm, this dicer enzyme complex recognize this miRNA precursor, which has this stem loop like structure. They recognize it, they cleave it and they form a small 20 to 25 nucleotide long miRNA molecule. This is how siRNA and miRNA reach to the cytoplasm. Now this miRNA and siRNA can either complex to RISC complex or RITS. What is RISC? RNA induced silencing complex. What is RITS? RNA induced transcription silencing complex. If these two siRNA and miRNA bind to the RNA induced silencing complex, this result in two things. First is the degradation of mRNA, and if the mRNA is somehow not degraded, they block the translation because the antisense RNA binds to the mRNA, so it cannot take part in the translation. Coming to the RITS, which is RNA induced transcription silencing complex, if this siRNA or miRNA bind to it, this complex, after degrading the sense RNA, this antisense RNA and RITS complex, they bind to the chromatin molecule. After binding to the chromatin molecule, they recruit certain enzymes. What they do? They methylate the histones proteins of the DNA. They degrade or they manipulate the protein. They modify the, not the protein but the DNA. They modify the DNA, because of which now the DNA cannot take part in the transcription. So if the siRNA and the miRNA complexes with RITS, this results in the blocking of transcription. So this mechanism is known as transcription silencing. In the subsequent classes, we will be talking about gene expression, gene knockout and the technique of gene silencing. So we will be talking about that in the subsequent lectures. Thank you very much.